Hello, everybody. My name is Brian, and today we will cover the topic of cell signaling. So, before we get into cell signaling, uh, let's just give it an overview of what it looks like. So, excuse me. So, as you see here, we have this membrane and we have this protein embedded with this membrane, and that will be the receptor. And the receptor will get a messenger or a ligand, as some will say. And this ligand messenger will be known as the first messenger because it's the first messenger that it's the first messenger of the process. Now what happens that we have a response by the receptor, and it's gonna create this cascade of membrane events that here. Now what happens sometimes in these what does happen in these membrane events that we do get the release of second messengers, as you see here, and these second messengers also bind to something else, and eventually they target the proteins that they have to target. Now, if we look back, we have extracellular events, so events that happen in the membrane. Then we have the membrane events that that happens as soon as the ligand is bound. Then we have these intracellular events that happen deep, and you see that the first part we call reception because it's receiving the messenger. The second is transduction because you see all these proteins are basically just these proteins are binding to another, or each protein is activating the next protein to transduce the message downstream. Then we have the cell response. So basically, after the message is transduced, it leads to the response that the cell wanted the messenger to do. And as you see here, these are the type of examples of proteins that it attacks. You see that they could attack transcription factors, which is used to regulate genes or to shut down a gene. It could be used for metabolic enzymes. It could be used for cytoskeletal proteins or transport proteins. There's many more, but those are just some examples of it. So we're gonna take a close look at hormones. So hormones are usually the first messengers. They'll classify in two ways. They'll classify by structure. So those amino acid hormones such as epinephrine, those peptide hormones such as insulin and glucagon, and those steroid hormones such as testosterone or estrogen. They're also classified by the mechanism. So some hormones or first messengers are called endocrine, which means that they act on distal cells. So like cells that are far away from the release, such as insulin or glucagon. Those autocrine that the messenger acts on the same cell, such as growth hormones, and those paracrine, uh, which means they act on nearby cells, so they don't go too far. That's gonna go like to the body cells and open the door. And these are like examples of these are neurotransmitters. Then secondary messengers. So secondary messengers are intercellular hormones of proteins or ions that are activated as by well activated by the events caused by the first messenger. So you see you have the first messenger goes to the membrane. It does the transduction with the membranes. Then you see the yellow highlight. These are some examples of first my bad, of second messengers. We do have CAMP, CGMP, IP3, DAG, calcium, and the process for these secondary messengers is to facilitate signal amplification. So they're just gonna bind to something else to transduce the signal downstream, more downstream, until eventually it leads to the cell response. So these are just examples which we will touch on in a bit. So the one certainly we are going to talk about is the G protein coupled receptor, or the GPCR protein. Now this protein is really abundant in our body. It's, I think there's over 1,000 genes or 10,000 genes that codes for different type of G protein receptors. So basically how these receptors work, you see the G protein will be the blue one, this blue one right here, and basically let me change the color. I don't like it. And basically what happens, you have this ligand or the first messenger that causes a conformation of change. So you see here, it changes this little bottom thing here. So basically the reason it changes it, so this thing We call it the G alpha beta gamma GTP subunit. So it's all these proteins in the GTP combined. So once the conformational change occurs, this sub this unit could bind to this, and it causes the first events of the cell signaling. For example, it could so as soon as this thing binds, we have something called the GTP or GGP exchange, which means that GTP is swapped out and GGP comes in to bind where GTP was once at. And you see what happens when GTP binds to this, it attaches to the alpha subunit and it separates the alpha subunit from the gamma, beta, and gamma subunit. So now you have the G beta gamma subunit and you have the G alpha GGP subunit. Basically, the G beta gamma subunit does its own cascade events, and the G alpha GGP subunit has its own cascade events. And for the G beta gamma, you see it stimulates phospholipase A, regulate ion channels, and regulate receptor kinases. So basically, those are some of the things that it's responsible for. But what we're going to focus more closely on is the activation of adenylate cyclase and the stimulation of phospholipase from the G alpha GGP subunit. So first, we're gonna look at the activation of adenylate cyclase right here. So this is an example of a glucagon receptor. So the glucagon will be the first messenger in this signaling pathway, or the first ligand that will bind to the G to this subunit. So basically, what happens? The GAGGP activates adenylate kinase right here, and basically the job of adenylate cyclase is to turn ATP into CAMP. And now just remember, as we touched earlier, the CAMP is the secondary messenger. So what does CAMP do? The CAMP has an important job. It's going to activate the protein kinase A. Now, protein kinase A and this glucagon receptor has a lot of functions. It could shut down glucagon synthesis. It could activate glycogen disintegration, and it could activate glucose synthesis. As we remember, glucagon was the first messenger here, and glucagon, the purpose of glucagon is or the reason why the body will release glucagon is to increase glucose export, because we're low on glucose, so it wants the body to release more glucose from its stores or to synthesize it from what we have already in our body. Now, as we said earlier, the gamma alpha GGP subunit has another function, which is to stimulate phospholipase C. And right here, and this is an example of the A1 adrenergic receptor, which uses epinephrine as a ligand. So epinephrine would be the one to bind to this protein to cause the change. So when it separates, the GAGGP is gonna activate phospholipase C. Now the phospholipase C is gonna activate PIP2 right here. Now PIP2 is hydrolyzed with the water, you see, and it creates two products. It creates the DAG, the DAG, and the IP3. Now the IP3, these two products do two different things, but they end up doing the same thing for the cell. So the DAG it straight up activates PKC or protein kinase C, and that it shuts down glycogen synthesis. And the IP3 is going to release calcium ions from the endoplasmic reticulum, and this could do two things. The calcium could help shut down the glycogen synthesis, and it activates that glycogen integration to release glucose. So this is another pathway that helps release glucose and help increase our glucose uptake when we are low, or when our blood glucose sugars are low. That's pretty much it for the signaling. As you see, it's not that complicated. It's basically just learning the receptors, the pathways, and what proteins and what secondary messengers go where, and where they bind, and what's the process of it. Thank you.